Hi everyone, sorry about those technical difficulties.
We're back. Did you put the donut in?
I did.
Hi everyone. Thank you for
Coming back.
bearing with us. Meredith, let's just wait for some people to join before we bring you on. Or should we just try it?
I think we should just try it.
Alright, Meredith, we're getting you on here.
Awesome.
Thank you
Thanks,
guys.
Annie.
Thanks for bearing with us. Alright, Meredith is on.
Okay,
Let's see.
how's it look?
Oh no.
Okay, it's pretty fuzzy, but as
Let's
long
just
as
go
we can
ahead.
hear you, yeah, we should.
Yeah, just go
Okay.
ahead, and maybe when I get off, it'll clear up. Okay, so I'll just kick us off. Hi, my name is Meredith Elliott. I'm a senior scientist here at Point Blue Conservation Science. I'm here at the headquarters in Petaluma today. In a minute, I'm going to pass it off to Olivia and Rebecca, who work in the Point Blue Marine Lab, which is just downstairs from me here in Petaluma. And they're going to show you some of the science we do in the lab and how it contributes to protecting and conserving nature. Under normal circumstances, I would be in the lab with these ladies, and we would have volunteers that would help us collect all this important data. But unfortunately, due to safety precautions around COVID-19, our headquarters will remain closed to the public for the time being. We hope, though, that through these virtual visits, which we plan to do more of, we can continue to stay connected with you, our community, and invite even more people to get excited about science and conservation. So first, we want to let you know a little bit about how it has been to work under these COVID-19 conditions, and assure you that we are being safe. So our biologists, Olivia and Rebecca, have been living together in our intern house in Petaluma, and they work together in the lab. So they're kind of like a family unit, and they have been able to continue to work through these shelter-in-place orders. A limited number of staff like me are visiting the lab to support the interns and following multiple safety precautions, including wearing a mask and being physically distanced when we meet in person. So although we know our data is important, we are prioritizing and monitoring human safety first. Okay, so let's get exploring to some marine science. I'm going to hand it off to Olivia and Rebecca.
Thank you, Meredith.
All right.
Meredith.
Hi everyone, thanks for joining. I'm Olivia.
And I'm Rebecca.
And welcome to the Marine Lab. So the Marine Lab's been running since 2006, and since we started, we've had about 30 full-time research assistants like us working in this lab. And we've been here for about a year. And usually during the summers, the lab is full of volunteers that are just looking to gain experience and see what it's like to be a marine biologist. But unfortunately, due to COVID-19, we're not allowed to have volunteers right now, but we're looking forward to when we can bring those back on. Oh, awesome.
Okay, we're glad for the videos.
Yeah.
Thanks for tuning in.
Yeah, thanks for commenting, guys. Yeah, so in this lab, we study marine food webs. So we're looking at the diets of marine seabirds, and we're also looking at zooplankton from our access cruises. So we get those samples by targeting krill. So we take like a big net and we scoop up a swarm of krill and see what species are present. But yeah, basically the work we do in this lab is to look at kind of who's eating who in the marine world and kind of trying to track these changes over time, because kind of the species we're finding. We can determine like breeding success, and we can also, see how these changes over time might affect the future. And with that, I'm going to turn it over to Rebecca to talk about our marine predator we're going to be looking at today.
Yeah, thank you, Olivia. So today we'll be focusing on the California least tern. Least terns as a whole are the smallest tern species in North America, but we will be focusing on the California least tern. I have this little decoy to give you guys a general idea of the size and what they look like. They're about the size of an American robin, if you know your birds. But yeah, so these are a West Coast subspecies in California, as their name implies, and they're actually endangered. So some of the threats posed on them are nest disturbances, predation, and habitat loss. But some conservation efforts that are being implemented are signage at nesting sites, habitat enhancement, fencing, and overall restrictive access on beaches that they are nesting. So these birds, they really like open beaches with very little to no vegetation. Like I mentioned, predation is a big issue during these breeding colonies on these beaches, so they want to camouflage it with the sand as much as possible. Their little chicks are very tan, and they have little black dots on the head, so when they are in the nest, which is not much of a nest, it's just a scrape in the sand, they can camouflage very well. But yeah, they nest here on the coast of California in the summer, and then in the winter they migrate to southern and central South America. But while they're here, we do some studies and surveys at these breeding colonies in California. And one in particular that we will be focusing on today is our least tern dropped fish study. So unlike other seabirds, California least terns bring back whole fish to their chicks and their mates. Other seabirds like to go and forage at the ocean, digest what they foraged, and bring it back to the breeding colonies to regurgitate to their chicks. But not these guys. They will go forage for fish and bring back the whole live fish. But sometimes these fish get dropped. This can be accidental. There can be another adult trying to swoop in and steal the fish, and it gets dropped. But basically, once these fish are dropped, they will not be eaten by an adult or a chick. Once they are on the ground, they're done. So field biologists that are conducting nest surveys at these breeding colonies will pick up these dried, dropped fish, put them in a bag, and bring it to the lab for us to further analyze. So we will be discussing that project with you guys today, and if you're interested, tune in. And if you have any comments, concerns, questions, feel free to leave them in the comments, and we will try to get to those as we show you the process.
Yeah, bear with our technical difficulties. Sorry, sometimes it might get grainy as we're moving around. Just let us know if you need us to repeat anything, and we'll do that. Yeah, and then also there should be a link attached to this video if you guys want to support our science. A lot of Point Blue's work is done because of donations from viewers like you. So yeah,
so let's get on into it.
So I'm gonna show you guys how we get our samples, and then we first clean them up.
Because they're very full of sand and nasty.
Yeah, and they're really smelly, so we wear gloves, otherwise we'll smell like fish all day. Yeah, so these are some fish that were picked up at our Hayward site. If you want to talk more about that.
Yeah, so we study a variety of breeding colonies along the coast of California, ranging from Northern California to Southern California. But this one is pretty north at Hayward.
You see this. Here's our fish. Yeah, and they're covered in gravel, as you can maybe see. So I'm just going to be cleaning them in the background.
Yeah, and And usually, this is a very small sample of ours. Usually we have very large bags. They drop a lot of fish, so kind of wasteful. But yeah, this is a small little sample that we have. We have our first question. How many fish will terns usually bring back to the colony in a single trip? Good question, Casey. Usually they have a very small beak, as you saw with this little decoy. So, but some of the fish are pretty small, so I think they can carry quite a few if it's a small fish. But if it's a large fish, it's probably just fat. And they are central foragers, so they breed in colonies that are very close to their food source. So they just go get the fish, bring it back, and go again. They don't waste any time. So they probably try to get as much as they can, but if not, they're very close to their feeding site, so they can just go back and get some more fish for their babies.
Yeah, and then, it's okay. Usually we get fish, but actually in this sample, I don't know if you guys can see, we have a little shrimp. You know, they'll get whatever they can find, and so in this case, one brought back a little shrimp and dropped it on the ground.
It's exciting.
Cute. Yeah.
Yeah, their food source is primarily fish, but they will often pick up little shrimpies or even octopus and squid and sometimes insects. So they are opportunistic. Let's see what other questions we have going. Quinn, do you know what kind of fish these are? Yeah, so once these are all cooked, and we'll get to that. Once we're done cleaning them, we actually identify what fish these are. And we will tell you how we do that.
Let's see if I can find. So this is your typical silver side. We'll talk more about these fish, but in this sample we have, it's like silver sides, and then we got a little goby here. And then we're getting a lot of these little killifish.
Mm-hmm.
So I think these are usually found in brackish waters. So this bird brought back one of these little killifish.
Yeah.
So these are done being washed. I don't know if you guys can see. So we cleaned off all the gravel, and now we're gonna put these in one of these pie tins, and we're gonna bake them. So.
And we do this just to prevent gross, mushy fish in our samples. And yeah.
And actually, yeah, preserves them. Some of our samples are preserved in like alcohol. We just dry these. They stay pretty good.
Yeah.
So we're gonna put it in our oven.
Is it on you?
Okay, so those are gonna dry. We usually do it at like 15-minute increments so they don't get too stuck to the pan. So we'll come back to those.
But as we wait, we'll answer more questions and show you some samples that have already been baked.
Any questions?
Let's check the comments. Thank you so much for joining, everyone. We got a lot of people. Thank you, Meredith, for answering some questions for us.
I think these are actually from our Hayward site. Usually they're from Alameda. But
Yeah, these big bags that we showed, these are from our Alameda colony right here in San Francisco.
Alameda, but yeah, so we're gonna talk about some of the species we find. You guys see this guy? It's a surf perch. And does anyone have an idea why these might get dropped on the ground versus, say, one of these guys?
And while we're waiting for your answers, I think we got a question. What is the strangest animal you found in one of these samples?
The shrimp are always fun to find, yeah.
Yeah. In these samples specifically, it's mainly fish, but we also study their diet in other ways and through their fecal samples, which we will probably do a live on in the future. And we find some fun stuff in those samples, so look out for that.
Oh, we did find a 3-spine stickleback, so that was cool.
Yeah. It's fun to get some variety.
Yeah. I'm talking about why they can't.
So does anyone have any idea about these guys in particular? Why they get dropped? Can kind of help you infer. See that beak? This is a very wide fish.
Just imagine this little baby trying to eat it. Yeah. Sometimes it can't really fit in the mouth.
Yeah, depending. Yeah, exactly, Emily. The shape.
Yeah. Oh no. Too wide, yeah. They're too wide.
Sometimes the chicks are not big enough and they can't really fit in the beak. But older chicks do like the cirque perch. Good job, you guys. You guys are smart.
This is our cirque perch. These are one of my cirque perch.
Really cute.
Then another one we find. This is a goat. Its eyes are kind of facing up. Oh no.
Technical difficulties. Yeah, their eyes are actually on the top of their head. But just keep going.
Okay, yeah. So these guys actually live on the bottom of the ocean. And so that's why their eyes are always looking up. That makes sense. But this can tell us that these birds live within an area that has gobies. So like mud flats have a lot of gobies. So we can kind of use that as to see like what kinds of species are in the area.
Right.
What is this, Rebecca?
That is a salmon.
Yeah.
Chin hook.
Yeah, baby salmon. We get those.
See some more questions coming in. Lishka, how long have you both been doing this study and what is the most fascinating parts of it for you? Michelle also asked, what's the most exciting part of your job? The study has been going on,
I think since the lab started. Maybe.
2006 or around that time.
Yeah.
So we have a pretty large data set to look back on and see how their diet has been changing, and that also shows us how the prey populations have been changing. Yeah, it's a pretty fascinating project. I guess the most shocking for me was the amount of fish that get dropped. I mean, this is a lot of food that is kind of being wasted, but at least it's being used for science now.
I think, the most fascinating part for me is just to see like the variety of fish that these birds go after, and it's pretty incredible that each one of these, to make its way into our lab had to be, picked from the ocean, meant to be fed for a chick, got dropped, and then scientists came pick it up, and then it got brought back to us where we do the rest. So it's just kind of a long journey for this fish, which I always find cool.
Right, exactly. Any other questions?
We got, where do you learn fish ID? Well,
sometimes people learn it in school, but we learn it here.
Yeah. We have a protocol that Meredith put together that has our most common fish that we'll find, but every now and then we find a different species, and it's always fun to try to figure out what that is. It's a little harder because when they're dried up, they're a little harder to tell what they are, because a lot of things that you look for, like fish, or like the the fin like length or like
other
like
small
things
factors
that get dried out that we can't really see.
Yeah, but it's a fun challenge. We have like a dichotomous key we can use and just kind of narrow down what fish, and based on like the range of that fish, that can really help us, and because we know where these fish are coming from. So if we can just like connect the two, we can be like, that is a goby.
Yeah. Yeah. I guess we'll show you some other fish. This is an anchovy. And this is pretty big compared to this guy, the baby anchovy.
Yeah, different life stages.
Yeah. Can you guess which one these little chicks prefer? Yeah, we'll wait for your comments.
We're getting a lot of questions.
Yeah,
good questions, you guys.
Yeah, thanks, guys.
Thank you, Meredith, also for chiming in on those questions.
Mm-hmm. Why is it important to study the diet of least terns? What's the bigger picture, or what bigger picture things can tell us about the ocean?
Yeah, we kind of touched on this, but we can go into more detail. Studying these marine predators not only helps us understand breeding success and the productivity of these birds as individuals, but it can really give us a bigger picture of ocean health, because these birds are firsthand studying the ocean. You know, they have specific fish species that they enjoy foraging, and some that they kind of resort to when their top fish is low in numbers. And doing the study year after year, we can really see how those fish populations are changing, and what species is more abundant, what species is like not there. Yeah, if you want to touch more on that. But yeah, just overall ocean health, it really gives us a good idea of fish populations.
Yeah, definitely. Yeah, and since they are endangered birds, it's important to know kind of what they're eating and what needs to be protected,
right.
You know, outside of just the birds themselves.
Mm-hmm. Good question.
Good question.
It's all about the big picture.
Yeah. Lishka has a question. What makes the adult terns drop the fish besides size and shape? Or is that the only factor?
Size and shape are usually when the chick is like, I can't eat that, Mom. Why would you bring that? But other reasons the adult can drop it is just accidentally. At tern colonies, it's pretty crazy. There's just adults flying everywhere. So they can also maybe run into another adult coming into the colony. Or, you know, these birds are sneaky. They might see a bird with a lot of fish in their mouth, and they try to come and steal it for their own chick. And that can cause dropping as well. So there's many factors.
Good question, Lishka.
Yeah.
Yeah, so Quinn said, back to our anchovy question, Quinn said, I would guess the chicks prefer the baby anchovies. Yeah, generally the babies are better for them.
Yeah,
yeah, they're just the right size. And anchovy tend to be fattier fish. This is actually pretty good for the chicks when they're first starting out, to be eating these fattier ones so they can put on more weight.
Mm-hmm.
But
when they're small, they like those small guys.
Yeah.
So.
We have another fish. They all kind of look the same from you, but we can tell them apart. This is Pacific herring.
Mm-hmm. And we'll get more into this, but one of the ways that we identify some fish that look very similar is by their teeth. Fish have teeth, you guys. It's kind of amazing. Yeah. But that fish in particular, the Pacific herring, the teeth is it's not where our teeth is. It's like on the roof of its mouth, which is very interesting. Why do you think that is?
Yeah, good question. Yeah, and then kind of the last group of fish that we're going to talk about are they're called silver sides. And I don't know if you can tell, but they're different sizes, but they can be like the same size, but they look very similar. And so we have to look at their teeth, like Rebecca was saying, to tell them apart. But Yeah, usually what we find mostly are these silver sides.
The bulk of this
I
bag.
would say, yeah, like at least 80% are these silver sides. And so when we are sorting through, we kind of put them aside so we can look at their teeth under a microscope. And yeah, we'll show you that now.
Should we check on our fish? Do you think they're
Oh,
fine?
I think, yeah, a little bit longer.
Okay. So we're going to show you how we identify by teeth. We actually have this really nifty microscope attachment that we will put the phone on. Sorry if it gets a little grainy. It should lighten up.
Let's let it, like, hey. Yeah.
So yeah, this is the mouth of a fish.
Yeah, I can zoom out.
There's his little face.
You see the fish? This is one of the fish we were just showing you guys.
One of the
Yeah.
So it could either
So
be a top smell, a jack smell, or even a grunion. Those are all same pan.
Yeah.
But yeah, we focus in on that teeth.
Yeah. So you can kind of see in this picture where the ends of the teeth are kind of forked. And so this is actually a top smelt. And these top smelt have a single row of forked teeth.
Yes.
Which is going to be a little different from the next species. Did everyone get a good look at that? Let's kind of flip them over. Yeah, sorry about the video quality. Let's see. Oh, there you go. Okay. Yeah, you can see those forked teeth right there. Pretty good.
Yeah, I'll hold that there.
Yeah. Yeah, so this is our top smelt. From what I've noticed, they tend to be like a little bigger than the top or the jack smelt.
Mm-hmm.
But yeah, so those are those forked teeth. And then we'll show you the one that looks nearly identical except for the teeth. This is going to be our jack smelt. Where are you? There you go. So
Yeah.
I don't know if you guys can see there, but jack smelt have 2 rows of needle-like teeth. So
there's no forking action going on in these guys. Just needle.
Yeah. Let's see, there you go. Yeah, some challenges with this though. Sometimes the faces are damaged and so their teeth aren't really present. So yeah, that's a hard part. And then also sometimes their like jaws are almost glued shut from the baking process. So we have to like pry open their mouths to see their teeth. But yeah.
Do you want to show the herring?
Yeah, we can show.
So now this is the Pacific herring. This is the one where the row of teeth is on the roof of the mouth. We'll try to show you guys. It's really cool. Maybe if we zoom out a little.
Yeah. Oh. We zoom down a little. You can kind of see in there.
Hard to see, but
yeah. So these differ from sardines in that these have teeth on the roof of the mouth, whereas sardines don't. But otherwise, they look the same too. So all these little bait fish kind of look similar until you really take a good look at them. Eyeball.
yeah, it's hard to see.
Yeah, you can't really tell. Sorry, guys.
But yeah.
Yeah, that's what we do. And then once we identify them to species, Rebecca can talk a little bit about what we do after that.
Yeah. So once we go through a big bag like that, we just kind kind of narrowed down what species we have with piles. So we have like an anchovy pile, a surf perch pile, goby, silversides, and all that. And then once we're done with that, we actually number them. This kind of lets us know how many there are. So these were already numbered. This is number 5. But yeah, we number them, and then we actually measure them. So we take the measurement from the nose to the end of the fin, and then so that's like the total length. And then we also do standard length, which is not including the fin.
Tail.
Or the tail, sorry. Yeah, this little tail.
Okay. So Michelle has a question. Which fish are the hardest to identify?
Definitely those silversides. I wouldn't say it's hard necessarily, but it does take the most time because there are huge piles of like, you know, silverside spa like species, and then we have to like go through, focus on those teeth, pry open mouths. Like, it just takes more time. I wouldn't necessarily call it difficult, but sometimes we get little outliers, I'd say, that maybe don't have teeth, or you know, like these herrings and sardines, maybe the vomer where those teeth would be is like not visible. So overall, we have a really good protocol made by Meredith to help us identify these guys.
Yeah, and some other things we can look at are also the scales, and like on the top of these fish there are 2 like top fins, and between them the number of scales between those 2 fins can vary between species. And then we can also look at their otoliths, which are their fish ear bones.
Right, there are many ways to identify these fish, surprisingly.
Yeah. But sometimes it's a little harder because, like, yeah, surf perch are pretty, you can distinguish them from the others, but some of these other ones are a little harder, especially when they're all crispy.
Yeah, that's probably the most like difficult, is when they're just, you know, kind of curled up and you can't measure them, or you can't really see those definiting factors. But overall, we identify the species. But yeah, once we measure them and number them, we weigh them on this little scale. Just pop them on. They're very light. And then we record it on this data sheet. So this shows us how many, the lengths. Oh, and we also take the depth. Totally forgot about that. And the weight and the species. And any notes. Sometimes any fish on the ground, no matter if there's tail or head. So, and we also take the width, depth.
Yeah, it's all great.
But yeah.
Okay.
Put back on the tripod.
Nice zooplankton guide.
So yeah, it's pretty much the process.
I think we might want to check on the fish real quick that are in the oven.
Oh yeah. Let's go see if they're crispy enough. We want them very crispy.
So you guys see that?
Very crispy.
So they're pretty crispy.
Oh no.
So yeah, we usually take a spatula and kind of make sure they're not sticking to the pan. And these little ones might be ready to go, but the bigger ones definitely need more time. Yeah, there's some fish blobs right there.
Yeah, some fish get all jumbled into a ball.
Yeah. Okay. So I think these bigger ones need a little bit longer, so we'll keep baking them for like 15 minutes until they're nice and crispy all the way through. I think we can answer some more questions.
Yeah. And you can imagine with a very big bag of fish, this can take all day trying to get these as crispy as possible. And we stink up the whole office.
Yeah.
So,
we usually have 4 pie tins going at once, and there's, a lot of fish that need to be cooked.
Yeah. This project has slowed down as breeding season has also slowed down. That's why our little sample is pretty small.
Yeah.
But yeah, these are from a couple months ago when breeding was in full effect.
So Lishka has a question. How does this internship fit into your overall personal career journey? You wanna go first? That's a good question.
Yeah, thank you, Lishka. Well, I definitely see a future in marine life, specifically seabirds. That is my passion. And this is kind of different than any other field experience I've gotten working with seabirds. This is like very, scientific, looking at lab work, really getting down to the numbers of the diet that they're eating and all that stuff. And I think that's really gonna benefit me as I pursue a higher education in wildlife. Definitely these lab techniques are gonna help me in the future.
Yeah, so she's more seabirds. I love the fish. So I think after this research assistantship is done, I plan to go to grad school to work with fisheries. I'm thinking about going to Oregon State University, which is awesome. But yeah, definitely the skills I've learned here are like incredible, and even connecting with viewers like you is really fun for me.
And a bunch of different biologists that also work here at Point Blue. There's a bunch of different projects happening.
It's so fascinating to see the span of like all the science is really great,
inspiring.
Yeah,
we're surrounded by very driven, smart scientists that are always willing to, help us and,
No, definitely my time here has been so wonderful. I really enjoy my job. Best job ever.
Good question, Lishka.
Yeah. Let's see.
Thank you again, Meredith. If your question was not answered, look out for Meredith's responses. She's very detailed with her answers, and she knows more than us.
Does anybody have any last questions for us? I think we're over our time by a little bit, but any last questions for the answer?
And if you know anyone that would be interested in watching this that couldn't make it to the live, it will be saved to the Point Blue Facebook page, so feel free to send it out.
Make sure you follow Point Blue on Facebook and other social media.
Yeah. I think we got another question.
What is the most surprising thing you have seen? In general,
or with the dropped fish? I guess with the dropped fish.
The drop
shrimp.
The shrimp, yeah.
Those are always fun.
Yeah.
Just the variety,
I'd say, in general here, like the zooplankton for me has always been so fascinating with all the different critters that come up. Not only the krill are in those net samples, we also get like amphipods and copepods and some things that look crazy, like out of this world. So stay tuned. Hopefully we'll do another live stream about the zooplankton project, which is probably one of my favorites. Just so many like weird things to look at under a microscope.
You never know what those nets are gonna catch. Michelle asked if there will be live streams in the future, and we hope. This one seemed to do very well. despite the technical difficulties. So yeah, we want to do one for basically every project that we do in this lab. So stay tuned for that.
Yeah.
Can you remind us why dropped fish studies are important? Yeah, so this study is very important because it gives us a general idea of how the ocean is doing based on these fish populations and what these birds are picking out there in the ocean and bringing back to their chicks, because they want the best for their chicks, so they're going to forage for the best fish out there. So we can really see that by counting each species, weighing them, seeing the different life stages out there during that time and how that's changing over the years. Pretty much all of the projects that we do here in the Marine Lab focus on ocean health. So we're really, because our oceans are changing, and it's important to know why and how, and kind of predict how it will change in the future as well.
Yeah. We have a question from Brent, aka my dad. Do you ever look to see what the fish are eating? We actually don't. That'd be a cool study. They're kind of dried, so their stomach contents are kind of dried inside them too.
But good idea.
Good idea. Yeah, it's always cool to see the food chain.
Yeah.
Yishka says, what's the next episode about from Life in the Lab?
We're thinking about staying with the least Tern theme. So, like I said, at these breeding colonies, we do various studies, and one in particular is our field biologists pick up poop samples that are around the colony, and also pellets, and bring them to the lab, and we look at them under a microscope and look for fish scales. Like Olivia mentioned earlier, that's another way we can identify the species. So we're hoping we can do that and show you some different scales. They're kind of like the fingerprints of fish. They're really cool.
Yeah, and kind of going off this is another question from Richard. Do you analyze the drop fish and or feces for presence of plastics?
Sometimes in our fecal samples, we do see some plastic fibers, and we note that in our notes. Yeah. But definitely not like the whole microplastics and all that, which would be a cool study.
Definitely.
We have all of past samples still, so if anyone wants to do that for a project, we have the samples. Okay. Group samples live would be very interesting. Thanks, Sarah.
Cool.
Yeah, so
yes, thanks for joining us today.
Yeah, we hope you guys learned something new and had fun. And be sure to check out that donate link, and also the careers page on the Point Blue website for future opportunities for you guys to maybe get involved like us. There are various positions here at Point Blue.
Yeah, definitely. And we're looking forward to, again, like our volunteers coming back. So if you're in the Petaluma area, like we would love, once it's safe to do so, we would love to have you guys come tour the lab and see what we do.
Yeah.
Great.
Yeah.
All right, thanks everybody for watching.
Thank you so much.
Yeah.
Bye.